welcome. Thanks for coming. I'm Richard Abelson, President and CEO of SDC. I, along with Dale Usner, who is CSO and Strategic Scientific Consulting Senior VP. Dale is a Ph.D. from Oregon State and has worked at SDC now for 8 years. And I founded SDC about 13 years ago. I have a Ph.D. in Quality and Reliability Engineering and a black belt in Six Sigma. So I find artificial intelligence and machine learning, innovations in clinical trial data automation to be a very interesting topic. Otherwise, I guess we wouldn't be speaking on it. And you guys are missing happy hour, so I guess you find it interesting too, so happy to have you here. Is it a revolution or evolution? Where are we at? You know, AI's spread all over this conference. There really wasn't anything last year, and now it's all over the place. And so what's it all about? If you understand, the history of statistical quality control and how that comes about, ultimately there was a Japanese post-war miracle where really Deming went over to Japan and helped kind of re-engineer how Japanese manufacturing worked. And ultimately, by the 70s or 80s, Japan was in many respects technologically more advanced than the United States. How did that come about? Well, it came about because there was a concentration on really Six Sigma and lean manufacturing as early techniques. They're called Six Sigma. Earlier it was, really just around statistical process control. And ultimately, this focus on quality and focus on reduced touch time, automation of repetitive tasks led to, less than a 30% increase in cost, since 1970 on an inflation-adjusted basis, compared to, 40% inflation, overall. So really, overall, the cost of vehicles that exist out in the world today just haven't increased that much in price over time. And this is because of automation. It's because of going from a period of time when all cars were manually built with your hands to a time when now machines are working on the line and engineers are working on the machines. And so the amount of energy put into the product was dramatically reduced. This drove higher quality data at a lower cost and less time. It led to higher ROI on R&D, increased profitability, and better patient care. This is where it's going to bring us. It's time for this industry. We have huge market forces that are driving us. We have a political structure that, whether it's Democrats or Republicans, everyone wants to pay less for medicine. So there's huge market forces at work that are going to drive down the cost of what people want to pay for drugs. That ultimately manifests itself in how do we bring drugs to market, faster? How do we bring drugs to market cheaper? Well, this is the way. This is how it's going to work, and this is, the path that we're going down. I think an interesting understanding, and I think there's a lot of misinformation about what is AI, it's sort of this mystical thing, making decisions for us. I think that a lot of people think that computers are out there writing code, and, obviously Elon Musk and what he advertises is, that's many, many years in the future, and that's not the reality of today. One of the things that you kind of close and personal is sort of automobiles. And so continuing on that trend is if you look at autonomous vehicles and how they describe what an autonomous vehicle does, right now, level 0 system warnings, we had that kind of for a long time. Level 1, that's what your car is mostly today, most of us, unless we own like a Tesla or a new Mercedes. And it's the driver and system shared control. As we go to level 2, that's hands off. The automated system starts taking control, but is closely monitored by the driver. So now, at this point, the car is turning the vehicle. The wheel will change. There's sort of some real, more advanced lane control. But you better be looking where you're going. You never know how the computer's going to, calculate what's going to happen. Ultimately, level 3, you can start looking away. The driver may do something else. The system will notify the driver if involvement is needed. Driver must be ready to intervene immediately. There's very few even cars out there now with billions of dollars being spent that are operating at this capacity. And level 4 and level 5, there's nothing out there that's doing that now. And there's nothing out there really doing that now in anything out here either. So it's a good understanding. So really now understanding that level 2 and level 3 now add a lot compared to level 0. But it's still not to a point where you're dealing with 100% control. So we're looking at what involvement is the driver having. At a level 3, really the driver's probably involved less than 20% of the time. Really the computer's doing 80% of the work, especially on a highway when it's especially controlled. So that's going to give us a lot. So if you start taking this and apply this as an approach towards how we can reasonably implement automation in practically in life today in our processes, then now you're saying, okay, it's not that the computer needs to do everything and we're not going to oversee it. It's not that we're going to interface with it. We're going to be meaningfully involved, and humans are going to have proper oversight of the computer in doing certain things. But mundane tasks are going to become more and more automated. And how do we get there? How do we merge things together in multiple data sets and some different things? There's some neat ideas. And so Dale's going to now present kind of a neat perspective, and I think one application that we've come up with that saves a lot of time, still requires human involvement, but is a meaningful process improvement that in an area that adds a lot of time to everyone's clinical trials. So I hope you enjoy it. Thank you.
Thank you, Richard, and thank you everybody for joining tonight. As I'm sure you've all seen, artificial intelligence is a key focus in most industries, and recently I've been seeing a lot of commercials and hearing a lot of commercials on how artificial intelligence and machine learning is essentially invading the adult beverage industry as well. So what I'm going to talk about today is artificial intelligence and the applications to data science in the pharmaceutical, biotech, and device industry. And so to start off, I'm going to go over some high-level definitions, just so that we're all on the same page. So artificial intelligence is a general term, and it's a term for any computer system that simulates intelligent behavior. This can be a large if-then statement, where every output is pre-programmed depending on the input. Most of us nowadays wouldn't think of that as artificial intelligence, but by definition it is. The computer is not learning, it's purely just outputting what was pre-programmed depending on the input. The next definition I'm going to go over is machine learning. Machine learning is a subset of artificial intelligence. And it's an application where the system actually auto-learns and improves based off of observed data. Finally, I'm going to talk about similarity models. And similarity models is purely a method for determining the similarity of two different textual variables. Now, applications in clinical trials. Some key focus points currently are on patient recruitment, where systems are built to comb through vast amounts of electronic health or medical record data, as well as the protocol of interest, in order to quickly identify patients that would be eligible for that protocol, vastly improving patient recruitment times. Next is patient compliance, where there are multiple strategies on patient compliance. One is to, very similar to the patient recruitment, understand based off of information what data, what subjects or patients are likely to comply with your protocol. Another is actually an application on your phone that watches you take your medication so that you know and the system knows that you're being compliant with taking of the medication. The third application in clinical trials is software as a medical device. This is frequently in a situation where you're looking to guide strategy for clinical or to diagnose a patient with a specific indication. And a focus point on this is image analysis, where you're inputting some sort of an image and you're able to identify and diagnose a patient that has a specific disease. And finally, drug discovery, where we're looking to identify treatments that are targeted to the specific biologics of different diseases. What we're going to talk about today is applications to the clinical data sciences. And so 4 applications that SDC is currently working on are the following. The first is the development of the draft case report form driven solely off of the protocol. System is basically going in, scraping the protocol, looking at the schedule of events, understanding what case report forms are needed based off of that protocol, picking from a library based off of the indication and understanding what measures are needed for that indication to draft the case report form. The next is an application for trending of the trial data as well as key performance and quality indicators. And this is an application we're looking towards cleaning the data as the trial is ongoing, as well as possibly identifying sites that might need additional monitoring or additional training. The third application is on EDC user acceptance testing, where currently this is frequently done in a manual state where somebody is sitting in front of the computer testing the EDC system, entering values, seeing if the query is firing or not, and documenting that. We're looking to automate that right now, where the system would actually go in, understand the different measures that are being taken, understand the different queries, auto-generate the data, auto-enter the data, as well as then develop the output and the documentation of that user acceptance testing. And then finally, we're looking at SDTM mapping as well as SDTM annotated case report form generation. And this is the topic that I'm going to elucidate throughout the rest of my presentation. So SDTM is short for Standard Data Tabulation Module. It's the standard specification and format for submitting clinical data to the FDA. And essentially, it requires converting the format of the collected data, either on the EDC system or central lab or reading centers, as well as adding some structural variables to that data in order to submit in standard format to the FDA. It also requires a separate annotated case report form. So everybody might be familiar with the annotated case report form that allows one to understand the EDC's design setup. This is a separate annotated case report form to understand the SDTM mapping. These requirements in total increase the SAS programming resource time for any given study by about 20%, assuming that you're not already building in efficiencies from a sister study. So can AI and machine learning make this process automated or more efficient? What I'm going to walk through in the next number of slides is the process that we went through to build an artificial intelligence algorithm. So the first thing that we had to do is we had to define our training data sets. For any artificial intelligence, what you want to take a look at, and especially machine learning, you want to have a training set of data. And so what we looked at are recently completed studies where we had SDTM development already completed, so that we were able to take a look at the raw data as well as the completed SDTM data. We got that data in a specified format and available for the software that we used to complete the artificial intelligence. The next step really is to train the algorithm on the data that we chose. So there's two sets of training. One is to take a look at certain variables and train via machine learning. And another, in the next step, is to also take a look at the similarity models that I discussed as well to further refine the algorithm for prediction and mapping to SDTM. So I'm going to walk through the rest of this example with a pretty basic case report form setup, but it's hopefully a good example. It might look straightforward. It's not. On the left-hand side, we have a demographics page, well, both demographics pages, if you would, on both the left-hand and right-hand side. What I'm going to key on here is race. As you can see on the left-hand side, well, let's note overall. The black text is the actual case report form text. The blue text is the annotation for the EDC system. So it's the variable name behind the scenes for the EDC system. So what you can see on the left-hand side is race is collected as a single variable. You have one variable for the race component. And essentially what you're doing there is you're collecting the primary race that the subject identifies with. On the right-hand side, you can see race. We have separate variables for each race category. And this type of system allows one to capture all the races to which a patient identifies. So we knew and we understood that we needed to build a system that would work in either of these scenarios. So what were the complexities? And what are the deliverables? So first, let's take a look at the deliverables. This next page is the same case report form page, but essentially I added in the SDTM annotations. They are the green boxes with the red text. It's the SDTM annotations that are needed to submit to the agency at the end of the study. And so that's what we're looking to develop. We're looking to develop that as well as the SDTM data sets. So as you can see here, in the scenario where we collected race under one variable, it mapped to DM race. In a situation where we collected race as individual variables to allow a subject to self-identify to more than one race, all of those variables together mapped to DM.race. Let's see how the algorithm, what type of information the algorithm had to work with and what it did. So this slide basically is showing you the output to each of those variables. So on the left-hand side is the race variable where we're collecting race as a single variable. The measures are the actual races. So you can see here we have a white, Asian, white, 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 American Indian, Alaska Native, white, white, and black or African American. I'm going to jump over to the right side where you can see the SDTM mapped data. As you can see, it's very similar to what's collected on the left-hand side. The only difference is capitalization. So a machine learning algorithm can very quickly go into this data, see here's the raw data, here is the SDTM mapped variable data, and it can quickly understand that, okay, if I see a variable where the measures have white, American Indian, Alaska Native, black or African American, and Asian, then I need to map that into the dm.race variable. So pretty straightforward. It's just a capitalization issue. But how about the scenario where we're collecting data under each individual variable for each race? So this data, as I have here, is true and false. It can also be collected as 0, 1s, yeses, nos, anything of that nature, but non-informative in itself. What I have here, just to highlight, I have got true listed in bold red for those situations where the patient is the race being identified by the variable name. So in the leftmost variable is race American. That's actually short for American Indian or Alaska Native. So it's true in the row identical to the American Indian or Alaskan Native. Same for the second column, it's race Asian. So if a subject is Asian, it's going to be true where the subject is Asian, and so forth. So as you can imagine, there are going to be many variables that have true or false, yes, no, 0, 1. We're not going to easily be able to define a machine learning algorithm by itself to train trues and false to know that that should be mapped to the race variable. And in fact, it shouldn't always be. There are going to be other variables that have true and false as their measures that aren't race. So we should not key on true and false here to map to race. What we do key on, though, is the data set name as well as the variable name. There's important information there. So what you can see here is the variable names start out with race, pretty important information, and also has additional text that help identify what specific race we're talking about. So we built a similarity model in there to read in and understand basically the data set name as well as the variable names in order to help map to the appropriate variable. All right, so this next table is essentially the output that's given at this stage by the algorithm. You can pretty much neglect the left two columns. That's just there because we need to know the domain. That's the data set that is DM as well as the study ID. But if we key on the variable name, the SDTM variable name, the variable label, essentially this is the raw variable name. This is the variable name in SDTM that the algorithm is predicting or mapping the raw data to, and the corresponding label. The next column is the probability of match. And the last column is what method the model used. So what you can see here is under sex, it's generally pretty standard. Via SDTM, you can capture it as male, female, M or F. And so that matched 100% based off of both machine learning and similarity. And ethnicity I'm going to just quickly talk about, and then I'll jump to race. In the upper table, which corresponds to the left case report form, and I didn't highlight this, but there was a category in that ethnicity that's non-standard. And so what that did is that actually lowered the probability of match because there was an extra category there that's not standard to general clinical trials. And so it had a little bit lower of a match, whereas the ethnicity in the bottom example, which was the rightmost example, has a fairly high probability of match at 98%. I actually consider 89% a really high match as well, just to let you know. But that's 98% because it did not have that extra category. So let's take a look at race. So in the situation where race was a single variable, it used both, as you can see here, it used both machine learning as well as similarity. The machine learning purely just said, all right, I'm seeing a variable that contains white, Asian, African American, Alaskan, American Native. So I'm going to map, because of that, I know I need to map that to dm.race. The similarity really came into place understanding that the capitalization was different. But taking those similarity models and knowing, okay, it's still the same word, we're good to go. In the second example, where we had individual variable names for each race, it's still mapped to race, but the probability of match is 73%. Now, that's really around essentially these additional texts for each of the race names not matching identically in all cases to the categories of race identified in SDTM. So there's a little bit of a penalty for that. And as you can see, that model also only used similarity because there really was not an easy way to use machine learning on true-false data. So the next step that we did in this case is, all right, first we had this training data sets that we could use to move forward. The next thing that we can use actually is standard documentation on SDTM. So there's standard implementation guides as well as standard terminology. And so what we wanted to do is bring those two documents into play as well to help refine the algorithm and the prediction. And so as an example here, I'm going to take the scenario where race was collected as a single variable. And as essentially a table that you see here, it's a table that I gave before. The raw variables are on the left table here, where you see American Indian, Alaskan Native, Asian, Black, African American, and so forth. The right table, bordered in red, is the code list value from SDTM standard terminology. And so, as I said, this is a second set of information from which you can help refine your algorithm. And so what the algorithm takes into account, here's the raw data, here are the code list values. Via similarity, it knows which of those line up with one another, and the output mapped value is indeed the uppercased values of the raw data, so that we are matching precisely the code list values. Now, another thing that I don't go into here, but one needs to take into account is whether or not there's terminology called extensible on SDTM, and all that means is, can you add categories or not? And so if it's a list that's non-extensible and you have a category that's not in there, we're going to penalize for it because you shouldn't have it in there, and so therefore it's less likely to be a match. But if it's a code list that is extensible, then we would not penalize. All right, then the final output really are the SDTM data sets as well as the annotated case report form. So here's the same slide that I showed before. It's the annotated case report form. The key thing here is the algorithm has run. It's taken all the raw data, variable names. It's mapped to the SDTM variable names. Now what it's going to do is it's going to take the PDF of the annotated case report form, read it, look for each of these EDC annotated variable names, understand based off of the application that we just had where it maps to in SDTM, and build out the annotations. So, and this is actually a key step that would save about 20 hours, just this component where I'm annotating automatically instead of having a human go in and manually complete that annotation. All right, so this is the full mapping process again. Identify what data sets you're going to be looking at, pull them together, get them in a format that's easily readable and usable by the application that you're using to build your AI machine learning. Build your model, look at both machine learning as well as the similarity models. Read in external data sources, the standards. As if y'all work in data management or stats at all, you know those standards change, so we've got the ability to read in updated standards and ensure things are working correctly. And then give your outputs. All right, lessons learned. Things that we learned as we were walking through this SDTM mapping process. One is start focused. That's hard to do. But define a fairly homogeneous set of training data. And what I mean by that is the following. Take care to look at the data that has been coded to SDTM or mapped to SDTM in the most recent version, or possibly one version older. Because in the future, when you're using this application, you're going to be using it to map to the current version. So training it on older versions actually just adds complexities that are unnecessary. I would also consider the same EDC system. Now this depends, of course, on your place of work. For us, we generally have 2 or 3 EDC systems with which we primarily work and then multiple others. So we stuck to our primary EDC system in order to develop this in a faster method. It allowed us to account for about 70% of our studies right off the bat before building in the additional complexities of the other EDC system structures. And then, and finally, this one was actually pretty funny. If you have data sets where SDTM was built by a different company, I would look at those data sets and make sure they're similar to the SDTM data sets that you might build yourself. And the rationale is everybody thinks SDTM is standard, so it can't be that different. It can be. So I would just make sure that what you're training on is something a fairly consistent implementation of SDTM as well. And then to begin with at the very early stage, just choose a subset of domains, start looking at that, get that working, and then broaden to harder domains, and then finally broaden out to the subdomains. Another key finding is look beyond the actual results in the measure. As you saw in the race, where race was captured as individual variables, we had trues and falses. Really not meaningful data for you in this mapping exercise. So we needed to incorporate looking at the variable names and looking at the data sets to really hone in on where we needed to map that data. And that happens as well on, let's say, adverse event forms. If you want to talk a little bit more serious, adverse event forms where you're looking at AE SEER being yes or no, and then the breakdowns of why it's serious are all generally yes or no's as well. And so you're going to have a lot of yes-no variables in the adverse event form. Again, not informative as far as the actual results are concerned. So there as well, you're going to want to look at the variable names to help that mapping. And then finally, most data scientists right now, today, are coming from a different industry. Generally, they don't grow up in clinical trials at this point. They're going to be coming from manufacturing or another industry. So they're going to be learning our industry and our process. So I would definitely train that data scientist on the manual process. And 2 weeks later, if not before, train them again. Because they're the ones who are going to be making decisions when they're seeing something in that application on which way to turn. And so they need to understand that process really well. And in fact, I would go back later and make sure you train them again. Because you can't pick this stuff up in one shot. And you're going to get the best product possible if you keep and continue to train that person. All right, key takeaways. So just from the get-go, AI is any computer system that simulates intelligent behavior. What we are looking at very briefly and discussed very briefly today is patient recruitment, compliance, drug discovery, and clinical data. Really all with the aim of identifying drugs faster, identifying patients faster, getting through the clinical trials, both faster as well as with higher quality data at less cost. As Richard said, we do not need to go to a level 5 automation to see benefits of AI. Our SDTM automation I would categorize as a 2 or 3, yet it's going to save, as I said, about 20% of resource time. So small investments to get to a level 2 or 3 are very much well worth it. And you're going to see higher quality data with less time and less cost. Altogether, driving the overall shorter cycle times to meaningful therapeutics. If you would like to discuss more about our applications or any other way we could help you, please visit our booth at 1239. Thank you. And any questions? Yeah, thank you. So I'm going to answer that actually in 2 steps, but make sure I answer your question. And so I'll restate the question. Is there any validation involved in individual projects? So first, let's talk validation overall of the algorithm. So when you get your training data sets, what you would want to do, I would suggest breaking that up. I would say 75%, 25%, where the 75% is going to be on training the algorithm. And then the 25% is on validating that algorithm and see how that algorithm performs on data that has actually already been, in this case, mapped to SDTM. So that is a very key part of the process in general before essentially putting into production your algorithm. And then the question that you asked, I believe, more specifically was on a given study, what the validation would be. So what we're doing right now, presently, is we are using our algorithm to define and create the validation data set for SDTM and still following the standard process for the production as a first step. So right now, Right now, the validation is that we do have a manual person still completing the production version of our SDTM data sets. We are looking for in the future, once we continue to hone our algorithm, to not have validation on the SDTM side, or to have more strategic validation. And that would, and where we're looking at right now is based off of, the probability that I had showed earlier. And right now what we're doing is we're outputting those variables that have a probability of a certain value or less as key variables to take a look at. Thank you very much.